It's great to see folks trickling in. We're just going to give it a couple minutes before we get things started. Thank you. All right, looks like we've hit a nice steady number here. So I think we're going to go ahead and get things started. For any of the folks on the line who have colleagues attending or might be interested in distributing a recording of this webinar, you can always reach out to us at PathAI. We'd be happy to share a recording for not only this talk, but also all others in the webinar series upon request. So I'm very excited to kick things off for this month's edition of PathAI's Machine Learning webinar series. As folks are seeing on the calendar here, we've had two previous talks. First in January, providing a brief introduction to machine learning for digital pathology, and then most recently in February with a focus on graph neural networks. Today, we're really excited to have Ylaine on the line to walk us through best practices in performance testing and model evaluation. And we'll be following this up with our next talk in April, focusing on a novel machine learning and imaging technology in QMAI, and then in May in building more interpretive and explainable end-to-end models and added multiple instance learning. Before we kick things off, really just wanted to give folks a brief moment to walk through this slide. Generally, this webinar is intended solely to provide information to a general audience and is presented for general purposes only and should not be construed as professional or medical advice from PathAI or its affiliates. I'll just leave this on for another brief moment for folks to read through before we hand things off. All right, and so just as a brief reminder, you can leave questions throughout the Q&A function on Zoom. All questions will be handled at the end of the webinar, but please, as they arise, as Ylaine walks through the content, we'll be keeping track of all of them and bringing them up as needed at the end of the session, along with any other questions that might arise. And so with that, I'm really excited to welcome Ylaine, Principal Biomedical Data Science here at PathAI. These are efforts in applying and interpreting AI histopathology models really across a range of indications. I'll be moderating. My name is Ryan Leung, Director of Oncology Products here at PathAI. And we're excited, I think, to provide really exciting overview now in PathAI's methods for performance evaluation and testing for machine learning and digital pathology.
Thanks for that intro, Ryan. Yeah. Thank you. Okay, so I'm excited to talk to you today about performance evaluation and testing. So what I'm going to do is to first introduce how digital pathology products are evaluated. Second, to discuss main challenges and approaches, and this will be the majority of the talk, walking through these challenges and potential solutions. And then lastly, to identify some key considerations when you're assessing validation studies for digital pathology products. So first, to introduce how digital pathology products are evaluated. This is a basic outline of a typical digital pathology algorithm and its components. So we start from a whole slide image. This slide is scanned to produce that digital image. We then pass it through an artifact model to identify the usable tissue areas versus what's background or what shouldn't be evaluated on that slide. This artifact model, the usable tissue is then passed through a tissue model to identify various substances, as well as a cell model. And these might be combined at the end for a slide level scoring model. So the kind of slide level scores that might be produced could be a disease severity score. It might be presence or absence of abnormal tissue, or it could be a biomarker value. So there are algorithms where it's definitely important to evaluate this final slide level score because that's the end product of the algorithm. But it can also be important to evaluate these intermediate models, the artifact tissue or cell models, not only because they inform on what that final slide level score could be, but because they themselves might be outputs that the user can see. For example, if there is an overlay of the different tissue types visible on the slide, you want to make sure that this overlay is accurate. So how are these validated? The two main approaches here are qualitative or quantitative evaluation. So for qualitative evaluation of these submodel components, the pathologist could review an overlay and say, okay, does it look like these substances are being accurately called in this overlay? For the final output, this would be comparison of the model final score to a pathologist's interpretation of diagnosis based on that slide. And then for quantitative evaluation, which will be the focus of this talk, for some models, we can do pixel by pixel comparison of what manual trained annotators, these are pathologists, give on a slide versus the predictions by the model in those same areas. And then for quantitative evaluation of a slide level score, these would be your typical slide level performance metrics like AUC or positive predictive value or accuracy. There's also a third type of validation, which I won't get into here, which would be a clinical or biological validation. So this talk will mainly be focusing on the quantitative evaluation of the models themselves. I want to show an example of this kind of process. So this is an example from NASH. So on the left is an H&E slide image from a liver biopsy. So the first thing that the model does is do an artifact model segmentation. So black shows the background and the rest is a usable tissue. And then from this usable tissue segmentation, a tissue model and a cell model can be applied. That tissue model on top is showing these NASH relevant substances like steatosis, ballooning, inflammation. And then finally, these submodels are passed to the final scoring model that'll produce something like a final NAS score that's useful for the diagnosis. So what are the challenges to evaluating digital pathology models? I'm going to divide this into 3 sections and talk through each. The first is determining ground truth for benchmarking performance. This is really important because pathologists themselves might disagree on the diagnosis or the particular presence or extent of substances on an image. So given that disagreement, how do we determine ground truth and how do we benchmark performance of our digital pathology products? The second is, how do we deal with the scale of histopathology images? These are potentially billions of pixels large. They can be really complex. The models might be predicting many different types of substances. The typical approach here is sampling, and I'll walk through a couple different ways we can approach that. And then finally, how do we design tests of our models so that we can estimate the likelihood of the product succeeding in the real world? Because at the end of the day, that's what we really want to feel confident about, is when we apply these digital pathology products to a new situation, that it's going to perform well, and that our test set is going to be informative of how well it's performing in the real world. So first, to walk through the first one, benchmarking against pathologists, let me outline what the issues are here. So digital pathology products are often benchmarked by comparing to manual pathologist annotations. And the ideal case here is if we have some sort of gold standard ground truth, that we know exactly what that slide level score is, that we know exactly what those substances are and where they should be within any part of the slide image. So if we have this sort of gold standard ground truth, we can compare our model against it. We can also compare benchmark pathologists against that same gold standard. And then we can see, okay, is the model any more or less accurate than these benchmark pathologists? And this will give us an idea of relative performance. Is the model just as good or perhaps better than pathologists at finding substances or doing a slide level score? But what's the issue is we often don't have a single gold standard source of ground truth. What's often done is instead forming a consensus of pathologist opinions. So if you have 3 pathologists, asking all 3 of them to try to agree or to try to take some sort of average across all 3 of their predictions. There are some problems here. So the first one is the difficulty in determining consensus, which I'll talk about next. And the second is that when you've formed a consensus with your benchmark panel of pathologists, and then you take that same benchmark panel and try to say, okay, how close is each of these individual pathologists to their consensus? It becomes an apples to oranges comparison when you're comparing the model against a consensus versus a pathologist who's already part of that consensus against that same group. Essentially, it's like a little unfair to the model because the pathologists in the model aren't being treated equally in this kind of relative performance benchmarking. So the second challenge is how do you get an apples to apples relative benchmarking? I'll also talk through one approach that we take at PathAI. So for the first issue, the difficulty in determining consensus, this is an example taken from a colonic biopsy from ulcerative colitis. So here are three frames, three small patches from a biopsy sample, annotated by 3 different pathologists. So you can see that there is some disagreement from pathologist to pathologist as to what the substances should be called. So for example, if we look at this lower structure over here, the first pathologist has identified some normal epithelium within this colonic crypt. The second pathologist has identified as a crypt abscess, whereas a third pathologist has identified this crypt as having infiltrated epithelium. So when we try to form a consensus here, it's really tough to decide, okay, which pathologist do we trust? How do we know if the model is close to the consensus, if there is no consensus to determine here? So what can we do? So the approach that we've taken to resolve these consensus issues is to do consensus-free benchmarking of the model against pathologists by iterative comparisons. So to walk through how this works, let's say we have our model and 3 different pathologists, A, B, and C. What we do is that we first compare the model against pathologist A by comparing the model against each of the other 2 pathologists as a reference. So we say, okay, compared to pathologists B and C, is the model closer to those pathologists? Is the model more in agreement with B and C than pathologist A? We also compare A against B and C. We then repeat the same procedure for pathologist B, holding A and C as references, and then pathologist C, holding A and B as references. So at the end, we can average those results from comparing model against A, model against B, and model against C, and have an apples to apples relative performance benchmarking. So this answers the question whether the model on average outperforms each pathologist in the benchmark panel when evaluated against all the other pathologists. So to move on to the second issue, which is the issue of sampling from slides, I want to show another example here. So this is an H&E stained prostate sample. And these samples can be very large and the model annotation is very complex. So when we look at what the model is calling as cancer versus stroma on the left here, or here's like a very small blow up as to what the cell annotations are for this model, you can see that the tissues are just very highly intercalated. And the cells, there's a lot of different types that are annotated and they're quite dense as well. We've estimated that manually annotating a single slide like this for a pathologist would take over a week for complete tissue annotation, or over a month for complete cell annotation. So from that perspective, it's very impractical to do exhaustive slide-wide annotations. And the approach that we take, and I think others have taken as well, is to instead use sampling. So to sample small sub-regions of the slides, acquire pathologist annotations within those regions, and then compare the manual annotations to model predictions within the same regions. There's 2 approaches that we have taken to this sampling, and I'll lay them out here along with some pros and cons. So the first one is manual selection. So we have a pathologist look at these samples, these whole slide images, and then identify substances of interest. So here, the pathologist has identified melanoma, areas of cancer within a slide. And then we ask the model to evaluate within those pathologist selected annotations, what prediction is the model making in those areas. The second approach is that instead of having areas be manually selected, we randomly select small patches from the slides, which we call frames, and then we ask both the pathologist and the model to exhaustively predict or annotate within these frames. So the advantage of the first approach, the manual selection, is that it's easier to gather data on rare substances. We can just ask a pathologist, hey, look at this entire slide and find, say, macrophages, which are fairly rare. Give us some examples of macrophages, and then we ask our model. This is very difficult in the second approach, the random patches, because you're relying on you're relying on the random sampling of frames to happen to select enough examples of macrophages that you can evaluate the model on these. And it can also be lower cost to do this manual selection approach because you're not requiring an exhaustive manual annotation within a frame. The con of the manual selection is that there is some bias here. And that's because when pathologist is selecting examples of a substance, they tend to select clear examples. So examples where they're certain that is really cancer or that is really a macrophage. And they'll tend to not select areas of a slide that may be ambiguous. And in that way, you might not be able to capture how well the model is doing on these sorts of ambiguous substances. And that makes it hard to extrapolate this kind of validation to a slide level performance. It may only really be relevant for these very clear canonical examples of substances. Whereas in contrast, for the random patch or frame sampling approach, you can, using this approach, capture a really wide variety of slide regions, both very clear areas and very ambiguous areas. And that way it's easier to extrapolate how the model might be performing at the whole slide level. I'm going to walk you through an example of how this random frame sampling works. So here's an H&E slide from esophageal cancer. I'm going to blow up a little section here, and you can begin to see some structures and cells in this blow up, and then blow it up even more to an area where it's actually feasible to perform exhaustive annotations. This is about a 75 by 75 micron area. And here I'm showing what the pathologist annotated each cell as and what the model annotated each cell as. There's various different cell types: cancer fibroblast stromal cells, lymphocytes, neutrophils visible in this view. And what's nice about this exhaustive frames annotation approach is that we can get really granular, really detailed assessments. So here you can see that if you compare a cell on the left, this green cell on top, to this green cell on the right, that the pathologist and the model agreed that this was a lymphocyte. If you look at maybe the cell in the middle, you can see an instance of disagreement or confusion between the model and the pathologist. And then if you look here at this group of red cells in the bottom, both the pathologist and the model are annotating them as cancer. So you can see on a really cell by cell, prediction by prediction, or pixel by pixel level, where the model and the pathologist are in agreement. So repeating this procedure across many frames, we can form these confusion matrices. So what this is showing is the pathologist annotations down the rows versus the model predictions across the columns. And then within each entry in this matrix is the number of cells that satisfy those conditions. So down the diagonal here are the numbers of cells that agree in their classification. We'll take it as example these lymphocytes. So of the pathologist predicted lymphocytes, most of them were also predicted as lymphocytes by the model, which is good. And then the greatest source of confusion here, you can see over here or over here, of lymphocytes were with neutrophils or plasma cells. So those were where the model was tending to get confused about what's actually a lymphocyte versus another kind of immune cell. So finally, moving on to the third challenge, which is designing of test sets. So when we want to design test sets, we want to make sure that the model performance is going to be reflective of its performance in the real world. And there's three main things to consider here. The first is that the test data should be independent of the training data, and that's in order to properly assess generalizability of results. If the model has already seen that image or even a very similar image from the same case, it might look like the model is performing really well, but you would be uncertain about whether it could perform well on new images or new cases. So it's important to make sure that all images in the test set have not been used in training. And you might go a step further and ask about, okay, what about new labs or tissues from new staining protocols that weren't exactly the same as what's in the training set? That can give you an idea of how well the algorithm might be performing on really novel data. The second is that the size of the test set should be sufficient for the statistical claims that are being made. And this is similar to an idea of powering your clinical trial. You want to make sure that if you're going to feel really comfortable saying that the model has 95% precision or 90% recall, that those confidence intervals aren't too large and that you feel comfortable making that claim. And my main suggestion here would be to simulate results under different kinds of acceptance criteria and assumptions of effect sizes, and to look at past data sets that were similar and see if you feel comfortable with those confidence bands. And lastly, and I'll dig into this one a little bit more, is that the test set distribution should adequately represent key subpopulations. And I'll go into what adequately represent means, but these key subpopulations should be defined by any covariates that are hypothesized to influence model performance. So ones that we have identified include technical covariates, such as stain or scanner or lab, and also biological covariates, such as disease severity, demographics, or disease subtype. So I said I'll get into what it means to adequately represent a population. So there are really different aims here and different ways that it can be interpreted for a test set to be adequately representative. So the aims here are transportability, balance, power, and difference, and I'll kind of walk through each of these in an example. So for the top tier, this is requiring subpopulations to be proportional to their representation in a target population for transportability. So for example, if you have a target population where you know there'll be about 4 biopsies to every resection slide, and you know that your test set you're aiming for is a size of 50 slides there, you should include 40 biopsies and 10 resections to be kind of representative of the proportions that you're expecting. The second aim is balance. This is to say population should be of equal size. So here, instead of doing 40-10, you would do 25 biopsies, 25 resections in order to balance out those two subpopulations. And you can see if you try for balance, maybe there'll be some trade-off in that you won't be able to achieve the same transportability and vice versa. The third way of adequate representation is for statistical power. So this is trying to ensure that each subpopulation is going to be statistically powered to pass some sort of acceptance criteria that you've previously defined. So in this example, if you know that you need an N of 50 to pass your AC, this would require 50 biopsies and 50 resections. So we're seeing that, okay, requiring power within each subgroup is going to require even more samples than just requiring transportability or balance. And then the final one requires even more data, and this is for an aim to detect differences. So if you want a comparison of subpopulations to be statistically powered, if you want to say that the model performs better in biopsies and resections, this will probably require even more than just merely powering for AC within each subgroup. So as you saw in this example, as you go down these aims, more and more data is required. So it's important to know at the beginning when you're defining your test set, which of these aims are you striving for? Just transportability, or are you going to require enough samples to detect a difference between subpopulations? So I wanted to wrap up by going through some considerations when assessing digital pathology validation studies and relate them back to what we discussed earlier. So first is to determine what part of the process is being tested. Are we starting from a glass slide or digital image? And then to make sure that your comparator, probably manual annotations, are being performed at the appropriate level, whether in the glass slide or at the digital whole slide image. And the second consideration here is whether you're evaluating a slide level score, the end product of an algorithm, or also the component submodels. The second main consideration is how pathologist variability is accounted for. How is ground truth determined? Is this voting? What will happen? How will ground truth be resolved if all the pathologists disagree with one another? And the second is to make sure if there's any sort of relative comparison of models against pathologists, that it's an apples to apples benchmarking, such as using this iterative pairwise approach that I outlined earlier. And then finally, knowing whether the test set's results will generalize to the future applications, understanding what approach was used to sample representative parts of slides. Is there a bias there, or is this an unbiased approach? And to make sure that subpopulations are adequately represented according to the aims of the study. So with that, I'm going to end and be happy to take any questions.
Awesome. Thank you, Ylaine. Looks like we have a few questions already in the Q&A. I think as we move through those, please welcome folks to continue dropping in questions. A reminder that the recording can be made available upon request in case there's anything you'd like to follow up on. Great. So maybe to start, I think a question that popped up really just when we kicked things off was generally how many pathologists are involved in collecting manual consensus annotations? Is there a hard rule, or are there guidance principles?
Yeah, that's a good question, and it'll depend on how variable that consensus is and versus how ambiguous the determination is. So the more ambiguous the determination, the more pathologists are going to be required to feel really comfortable that you're representing the spectrum of opinions, and that any average across those pathologists is going to be accurate. So one thing you could do is to look at example data that was collected on with different numbers of pathologists, and see if do you get a different answer as to what the consensus is if you take out a pathologist from that set. So let's say you have an example data set that used 5 pathologists, try taking out 1 pathologist and recalculating consensus and seeing if that changed or if that remains stable. And if it seems really sensitive to the number of pathologists, that's an indication you need even more for a stable consensus. Whereas if it remained the same, that suggests that, okay, you were adequately covered. Maybe you could even go to fewer and still have a really stable and solid consensus.
Awesome. Really leads well, I think, into the second question, which was around pathologist disagreement. And so if pathologists are widely disagreeing about, in this context, the identity of a cell or a particular tissue region, how do you anticipate that would affect evaluation metrics? Are there ways for you to be able to determine that? And are there particular scenarios maybe where a lack of consensus is really a challenge?
Yeah, there definitely are situations. I think in general, if we think that there is just a moderate amount, we do recommend this iterative pairwise approach that I outlined. So here we avoid directly calculating a consensus, but instead asking whether the model agrees with each individual pathologist and quantifying that extent of agreement. So there you're just saying, okay, we're going to not try to say exactly what the right answer is, but just say on average, how close is the model performing to the pathologist, and trust that the closer a model in general performs to pathologists, the better.
Great. Are there other ways to test for model performance outside of, say, comparison to pathologist annotations or manual scoring methods?
Yeah, that's a great question. So one way is through positive. I'm going to probably talk about 3 ways. So one way is through positive controls. So if you, just like in a lab assay, if you have a sample of tissue that you know should contain that marker of interest, for example, PD-L1 using tonsil tissue, you can make sure that your model is correctly identifying the presence of that marker or of that substance on that slide. The second way is through an orthogonal method. For example, you can compare identification of certain cell types on an H&E slide with a paired MIF, the immunofluorescence or FISH slide, and see, okay, like when we specifically look for that cell marker and we do that on the same slide, do we get the same answer as when the model is trying to infer cell type just from H&E staining? And then the third way is what I didn't focus on so much in this talk, which is biological or clinical validation. So if you can show that what the model is predicting on the slide correlates with some sort of a biological outcome, then I think that's highly suggestive that what it was identifying was really relevant to the underlying disease biology.
Great. And I think actually touches on another question that had come in, which was a consideration towards outside of H&E using any special stains. So I think you had already just mentioned maybe IHC, multiplex immunofluorescence. Are there any challenges with those specialty stains that come to mind when you consider ground truth?
Yeah, I think this talk was fairly general. So these ideas of determining relative benchmarking of how you sample from slides, of performing these really granular comparisons will extrapolate well to these different staining approaches.
Great. Something that I thought you'd brought up that is also now relevant to this question was how you'd sort of use the evaluation methods to understand how models are performing or how you might improve them. I think especially when you were describing the confusion matrix. So the question was around how you can use evaluation methods to determine what might be driving underperformance for a model. Are there ways that you can leverage metrics to help improve performance results? So using the evaluation to not only benchmark your performance, but also inform how you might improve?
Yeah, that's a good question. So one thing that can be done with these really highly granular results like a confusion matrix is you can see what substances are confused with one another, and then to adjust your model training or your model inference to account for those confusions. So if you see that you're consistently under predicting cancer, can you add some more examples or a more diverse set of samples to your training so you can better represent that substance?
Awesome. Looks like those are most of the questions that have come in. I'll leave 1 to 2 more minutes maybe for any others. And otherwise, you can always reach out to us at PathAI with any follow-up. And again, the recording will be distributed in case any other thoughts come up in the meantime. So just give it another minute. All right. I think then we'll just about end there. Thank you again for joining this edition of PathAI's webinar series. We look forward to seeing you, I think, in the following months. We'll be back with an exciting talk around QMAI, as well as end-to-end models and ways to make them more interpretable. All right. Thank you.